Hi, this is Mia Baum. So today we will be talking about the movement of macromolecules via flowchart. So this is the complete flowchart. I will be discussing phospholipids, insulin, and actin. The processes are summarized in miniature flowcharts in the following slides. So first we're going to start with phospholipids. Phospholipids are essential components of cellular membranes. They contribute to membrane structure, fluidity, and signaling processes. Their synthesis begins with a ribosome in the cytoplasm. The ribosome transcripts and translates the genes specific to these phospholipids. This information then moves to the smooth endoplasmic reticulum, the primary location of phospholipid synthesis. Within the smooth ER, enzymes catalyze a condensation reaction between glycerol 3-phosphate and fatty acyl-CoA to form phosphatic acid. Phosphatic acid is a precursor to phospholipids. Once matured within the smooth ER, the phospholipids are moved to the Golgi apparatus via transport vesicles. Here, they undergo further modifications, specifically the addition of specific head groups and fatty acid chains. Once modified, the now complete phospholipids are packaged into secretory vesicles. Finally, the phospholipids are moved to the cell membrane for fusion. This fusion allows for phospholipids to be released into the extracellular space. This is a crucial process for maintaining lipid homeostasis, cellular signaling, and membrane biogenesis. The next process we'll talk about is insulin synthesis. Insulin is a peptide that is essential for glucose homeostasis. The synthesis of insulin occurs in the pancreatic beta cells. The synthesis begins in the ribosomes attached to the rough endoplasmic reticulum. Within the ribosome, transcription and translation of the insulin gene occur. From here, proproinsulin is processed by the rough ER and moved to the Golgi apparatus for further maturation. Once the Golgi apparatus obtains the insulin matures and is packaged into secretory vesicles. The vesicles move to the cell membrane and fuse there. This allows the release of insulin into the bloodstream. Once in the bloodstream, they can respond to stimuli and bind to insulin receptors. This process allows for the regulation of glucose uptake, glycogen synthesis, and various other metabolic processes. Actin synthesis and intracellular use is next. Actin is a globular protein that has many diverse roles and is a key component of the cytoskeleton and has roles in mobility and intracellular transport. In the synthesis of actin mainly occurs in the cytoplasm of the cell. Ribosomes in the cytoplasm transcribe and translate mRNA coding for the actin. The actin is then used in various intracellular functions. Monomers of actin assemble into filament structures by regulation of actin-binding proteins. This contributes to the organization of the cytoskeleton. The proteins also interact with motor proteins to generate contractile forces and further facilitate intracellular transportation. Actin additionally contributes to cell signaling pathways by interacting with regulatory proteins, which modulate cellular responses to extracellular stimuli.